Today we'll discuss the cell cycle. Having already talked about DNA and some of us chromosomes, the cell cycle is the next logical step. It's important to remember that all cells go through a specific life cycle called the cell cycle. Like anything, there's two main parts. There's interphase. Think of an interstate as a highway between two states. Interphase is the longest phase, and the most important thing that happens here is that the cell grows and it replicates its DNA. Another word for replicate might be duplicate or even copy. The second part of the cell cycle is cell division. This is where the cell, after having copied its DNA, actually divides from one parent cell into two daughter cells. And then two become four, those four become eight, and that's mitosis. There's another type of cell division called meiosis, but we'll save both of those specific types involving specific cells for our next unit, which will be next month. Cell division consists of, in this instance, mitosis and cytokinesis. So here's an overview of the cell cycle. Like any cycle, you can see you can start anywhere, just like our cell respiration and photosynthesis cycles. Doesn't matter where you start, you'll end up back there eventually. But let's start here. At the beginning of interphase, you go through a cell growth period because you just came out of cell division. So now these new cells have to grow. Then you go into an S phase. S stands for synthesis or synthesize. And what you're synthesizing is new DNA. See that right here, new DNA. Then G2 is where you actually grow again and do some final preparations for cell division. Here's another more broken down diagram illustrating the cell cycle. You can see there's G1, S, and G2, but now this M phase is broken down into more specific smaller phases, which we'll hopefully have time to get into. That's PMAT or PMAT, prophase, metaphase, anaphase, and telophase. What's not in this one though is cytokinesis. Some people include that as part of cell division. Other people talk about it being a separate process from mitosis. I think it actually starts during telophase and ends up after telophase is complete, so I would think of it as part of actual cell division. So interphase, again, there's that G1 or gap or growth 1 phase. That's that where the cell grows before it actually copies its DNA. There's the S phase, notice it's big and bold, because we're going to talk about it in more detail presently. And then there's that G2, that's where you actually go through your final preparations for cell division. That involves a lot of your organelles and things like that. So the length of each part differs amongst different cell types. Some cells that are going to divide more quickly, they'll have a shorter interphase. Other cells that once they're going to have a longer or indefinite interphase. So let's talk about DNA replication. First of all, you see the DNA has this classic double helix shape. We learned about that before. You see how because of the phosphate bonds, the sugar phosphate backbone, it actually goes in a nice helix pattern, tracing along this. Well, notice that these are color-coded. There's a red and a blue, and they're just wrapping around each other. We call these complementary strands. Think of the word complement, it means that something is going to match something else, such as a handbag or clutch complementing your shoes, or maybe a tie complementing my shirt, belt, and shoes today, something like that. So the first enzyme that comes in and uncoils this molecule is going to be a helicase enzyme. Think of helicase as like a helicopter. It goes around and it spins, and as it does so, it uncoils and unzips those hydrogen bonds. Those unzipping the hydrogen bonds of that DNA molecule can be done because these bonds are incredibly weak, which is a good thing. Think of it as like a Velcro shoe. You know, that Velcro strap on the top of those shoes are strong enough when it needs to be, but with minimal effort, you can pull it apart. After the helicase enzyme comes through and uncoils and unzips your DNA molecule, you have a DNA polymerase enzyme. So DNA, because that's what we're trying to make, polymerase, it ends in ASE, gives you a clue that it's an enzyme, and it's a polymer enzyme. A polymer is anything made of repeating subunits. So remember that DNA is made of A's, T's, G's, and C's. Those are nucleotides. Well, each of those nucleotides is a building block or a monomer that makes up a polymer. So DNA polymerase enzyme that moves along each individual strand one at a time, and it connects these complementary bases. Remember, complementary means they match. So the final product is two identical copies of that original DNA molecule. Again, let's go back to the complementary here, look inside. You've got a C, the complement of C is G. You have a T, the complement of T is A. So A goes with T, G goes with C. How often? Always often. Adenine always bonds with thymine, and guanine always bonds with cytosine. The sooner you can come up with a mnemonic on your own that's going to help you remember that, the better off you'll be. ATGC. I always remembered it as Aaron and Travis's grandma is cool, because she was, right? Again, with it being Halloween, maybe you'd like to think of all teens like a good candy. I don't know, that one's a little lame, but come up with your own. So once the DNA copies itself, let's go back to our chromosomes. Remember that a single strand of DNA, once copied, becomes two sister chromatids. See how they make this classic X shape? This should be review. Again, we talked about it earlier. So you've got one single strand of DNA here. Once it goes through DNA replication, where helicase uncoils and unzips it, breaks it apart, and after that it comes in with DNA polymerase and matches up on each strand complementary bases. Once that happens, now you have an X-shaped pattern, which are two sister chromatids, and notice they're attached at this centromere. The centromere is going to come into play during cell division, which is the other part of the cell cycle. So here's a nice illustration. Again, look, A, T, T, A, G, C, C, G, all the way through. The blues, your original strands. The enzyme helicase comes through and breaks these apart. And then DNA polymerase comes through. Now, granted, it does it differently on the different strands, the leading and the lagging strand, but we'll talk about that another day. We're trying to keep it simple and introductory level for now. It comes through and see it matching up these complementary bases with these other nucleotides here. So like GC, and then on the other side it takes C and matches it with G. On this side there's A, matches it with T. On the other side there's T, it matches it with A. So the final product is two exact copies. Now, barring any mutations, these should be identical. So let's look. AT, AT, and AT. TA, TA, and TA. That's exactly how that works. So you can see the blue strand is the old and the red strands are the new. They call this semi-conservative DNA replication. It means that half of each new molecule is actually half of that old molecule. So only half of the information is new each time. So again, semi-conservative DNA replication. Here are just more illustrations doing the same thing. So let's do one more thing. Come through right here. And sure enough, here's a DNA molecule. AT, TA, GC, CG, AT. So helicase is going to come through and split these apart. Now remember, can't really illustrate it here in Microsoft Word, but what it's doing is it's actually uncoiling it as it breaks those hydrogen bonds in between. So the next enzyme that comes in is DNA polymerase. So here's an A. Let's change colors here. It's going to pair it with a T. Here's a T. It's going to pair it with an A. Here's a G, C, C, G, and A, T, C, G. Pretty straightforward. Let's look at the other side. Here's a G, so it pairs with a C. Here's a T, so it pairs with an A. Here's a G, so it pairs with a C. Here's a C, so it pairs with a G. Here's an A for adenine, it's going to pair with a T for thymine. And here's a T, which pairs with an A. So once again, your final result is two identical copies of that original DNA molecule. AT, AT, TA, TA, GC, GC, CG, CG, AT, AT, CG, CG, just like that. The outside are your older strands, and the insides are your newer strands. Either way, now that you have two copies of your DNA molecule, you have a chromosome, and each copy represents one sister chromatid. The next stage of the cell cycle is going to be cell division, and that's when we actually rip these sister chromatids apart, and they each go to a new cell. We'll talk about that more